Hey everybody, today we're discussing Hardy-Wienberg genetic equilibrium. In 1908, the two scientists G. H. Hardy and W. Wienberg suggested a theory about evolution, and they said that the allele and genotype frequencies will remain constant from generation to generation in a population. There are 5 requirements for their theory. Their requirements are, here, let me get this out of the way. Their requirements are, 1, the breeding population has to be large, so, we gotta have a lot of things to mingle. Their second rule is that mating is completely random. That means these guys mate, and these guys mate, and these guys mate, and these guys mate. Well, there's too many in there. Anyway, it's completely random, just like me drawing circles. Their third law is that there were no mutation of alleles. That means and this is just for the sake of simplicity, this is one allele and this is the other allele. They said there was no mutation of alleles. Therefore, these alleles will be exactly the same in everyone that has those alleles. There is no mutation. Like when cells are replicating, they replicate exactly the same. They don't mutate or differ at all. The fourth rule is that there is no differential migration. So that means that nobody is coming into a population and nobody is going to leave the population. The fifth rule is that there is no selection. That means all genotypes have an equal chance of surviving. That means that having the A, the big A, little a, does not give you any advantage over having a little a, or a big A, big A. So before we start disproving Hardy-Wienberg genetic equilibrium theory, let's go over the math. You see this equation right here, p squared plus 2pq plus q squared equals 1. That is the genotype frequency. So that means that it represents the frequency of big A, little a, and little a, little a. It represents the frequency of each of those genotypes within a population. To review the math, I'm going to go through a practice problem with you. The practice problem is, in the United States, about 16% of the population is Rh negative. The allele for Rh negative is recessive to the allele for Rh positive. That means that the Rh negative allele is the little letter, or the little a. In this case, we're going to be using R's, but and then for positive, you just need to have one big A or double big A, or double big R's, I guess. And then it goes on to ask us, if the student population of a high school in the U.S. is 2,000, how many students could we expect for each of the 3 possible genotypes? So that means they're asking us to find how many students are big R, and how many students are big R, and how many students are little r. So that's what we're going to do. We're going to find that. So let's set up our equation. We know for a fact that 16% of the population is Rh negative, and the only people that are Rh negative, because the Rh negative allele is recessive, are people with the double little r's. So when we plug into our equation p squared plus 2pq plus q squared is going to be 0.16, or 16% of the population, because this represents this represents this, and this represents this. So what we're looking at is not actually q squared, or it's not q itself in place of q squared, it is q squared. So we need to find the square root of 0.16, which will be 0.4. And then you take that and you simply plug it into the equation for the allele frequency, which is p plus q is equal to 1. Now, in the place of q, we're going to be using 0.4, so you just transpose 0.4 over, subtract it from 1, and then you should get, for p, 0.6. Now you have p, not p squared, so you're going to take 0.6, and you're going to plug it in, and you're going to square it, and you should get 0.36. Now you have two things. You have p squared and you have q squared, and now all you have left to find is the frequency of the genotype big R, little r, which is 2pq. So just a sec, let me clean this up a little bit, and I will find that for you. Sorry, gotta erase some stuff. Okay, so now we're working with 0.36 plus 2pq plus 0.16, and it's really simple to do the 2pq thing, and you guys might find faster ways to do the math. I'm just doing the basic, simple. It's kind of the long way, but it gets the job done, and it's simple. And now you can just, when you're finding 2pq, you can just plug in the q and the p. So that means our equation is going to look like this to find 2pq. 0.6 for p and 0.4 for q. And when you multiply 0.6 and 0.4 times 2, you should come up with 0.48. And this is where you plug it in for 2pq. So your equation comes up to be, just a sec, I got to do some more cleaning. Your equation should come up to be 0.36 plus 0.48 plus 0.16, and this is going to equal 1. So now you know that your equation is correct, and you can now multiply the total population that we had earlier of the high school that we're trying to find, which is 2,000. You just multiply 2,000 by each of these spots to fill in what these genotype, the number of people with the genotypes are. So here you have the genotype frequencies, and you just multiply them by the actual population to find the total number of people that have that genotype. So I've already done the math up, so we don't have to go through it, but 0.36 times 2,000 is 320. 0.48 times 2,000, sorry, wrong part, my bad. No, 0.16 times 2,000 is going to be the 320. 0.48 times 2,000 is 960, and 0.36 times 2,000 is 720. So here we've got the number of people in the population that have these specific genotypes. So then we went on to, test the Hardy-Wienberg equilibrium theory. We created a case study where all of the 5 requirements were met, except, well, we used a smaller population simply because it really doesn't honestly matter that much, because you're still, the same thing goes on. It's just, it was easier to use a smaller population for us. Later we'll use a larger population. But the mating was completely random. There was no mutation of the alleles. There couldn't have been, because of the way in which we did it. We used playing cards that had A's drawn on them. So we had a big A and a little a. And we started out, everyone started out, all 6 or 7 of us, started out with a big A and a little a. And then we would just randomly throw down one of the cards to see which allele was passed on. So it was completely random and there was no mutation. there was no migration because our population didn't increase any or didn't decrease. We just simply moved on generation to generation. We kept going until we reached 5 generations. So we went through 5 generations. Oh, also there was no selection. In this lab there was no selection because each genotype had every possibility of surviving just as much as the other ones. So you can see in this picture we've got my initial phenotype was big A, little a, and so was everyone's. So the genotype frequency was 1. You can see that right there. Then what we did is we mated, and we did it for 5 generations, and after my ending genotype was little a, and so we plugged that in. We didn't have any big A's, but we plugged it into the equation, and we found the number of people that had each of these genotypes, and then we found the frequency. So our starting frequency of the alleles, P's starting frequency was 0.5, and Q's starting frequency was also 0.5. And as you can see, they differentiated, they changed, which right off the bat disproves Hardy-Weinberg equilibrium. And we followed all the rules, except for the breeding population is large, but I will demonstrate later how we actually did follow that. Here's our second case study, and we did this one where we changed it so that there is natural selection. The natural selection is that the little a allele had a susceptibility to a disease or whatever and died generally before the age of 3, therefore they could never grow up to survive. They just died. Those children died. And so that means that if say I was mating with someone and I threw down a little a and they threw down a little a, then what would have happened was that that child would have died, and we would have just gone again until we got a viable match. So I started out just like everyone else with a big A and a little a, and then through 5 generations I ended with the big A. So what it looked like, our frequencies changed. We started out with a frequency of 1 in the big A, little a, and because the negative was the dominant trait, the negative for that disease was the dominant trait, it became more powerful and obviously started showing up more. You see the big A's, and you can see the big A allele is P. So P started out as 0.5 and Q started out as 0.5, just like in the last test. But you can see how it changed. It ended up as a 0.833 and a 0.166 because the P, or the big A allele, was dominant and it was the negative for the disease, so it survived. It was the surviving allele. So here's case study 3. Similarly to case study 2, if you ran into a child with double little a's, they died immediately. However, we're trying to replicate such things like sickle cell allele in populations, so that means that the heterozygote, or the big A, little a, the heterozygous person, they have a greater chance of surviving, because if you're a homozygous for the dominant trait, there's a 50% chance of you dying, and then there's a 100% chance of you dying if you're the homozygous for the recessive. So if we ran into, when we were mating, if we ran into a child that had double big A's, we would flip a coin, and heads it would die, tails it would survive. So we went through our, we did 10 generations, and our starting frequencies were 0.5, 0.5, similarly to the other ones, and they changed to, the dominant did not as drastically change. You can see it from the last picture. It did not as drastically change because if you had the big A, there was a 50% chance of you dying. So the natural selection kind of honed in. It was like, yes, it's good to have the dominant trait, but if you have too much of the dominant trait, that can be bad too. So it kind of started to even it out more so than if, if it was one homozygous was terrible and the other homozygous was okay. It evened out the whole frequency a bit more. Not quite as much as in the first case where everyone survived, so there was not much change because it didn't really matter. Our final case study was exactly like what we did with the first one, except this time each student, there's 6 or 7 of us, each student had 5 people to make, or had 5 different, I guess you would call them identities that we were mating with. So it was a much larger population, and this was just to try and, help out with the Hardy-Weinberg thing. But in this one, there was no natural selection, there was no death of genotypes, no genotype favorability. So everyone started with big A and little a. So our starting frequencies were 0.5 and 0.5, and they came up to be 0.57 and 0.43, which was better than the small population, I believe. Yeah, see, there was a greater number of dominant genes, the big A's. There was a greater number of those than there was in the original. Basically what happened was they just swapped, the P and Q swapped from the bigger generations, but it still disproves Hardy-Weinberg, because if it was a true Hardy-Weinberg situation, the alleles would still be 0.5 and 0.5. So the whole point of this exercise was to go over the math, and I showed you the math, how to calculate allele frequencies and how to calculate genotype frequencies, and just to show you, to disprove Hardy-Weinberg, and cover the fact that it's not actually possible, because mutation occurs and natural selection does happen because certain alleles are weaker or susceptible to diseases and stuff like that. But it was a good principle, and we got good math from it. Great math came from their mishap. Just as a quick reminder, just to go through it all again, P represents the number of big A's, and Q represents the number of little a alleles that there are in a whole population, and they equal this percent frequencies, and you just plug that into the bigger equation to find the genotype frequencies, and it's actually all pretty simple. Now, this is something that I found funny when we were going over this. I was playing Minecraft late at night when I should have been doing my biology homework, and I came across the idea that in Minecraft, the genetics for the animals actually follow the Hardy-Weinberg principle. And I'm going to include later a video about that, just as a fun little thing. Hardy-Weinberg is by no means correct in the real world, where because there are just simply too many factors, and there is migration, and there is all these other things, mutation and natural selection. All of that does exist in the real world, so Hardy-Weinberg is not plausible in real life. However, in video game, it's very plausible, so I'm going to include a fun little video about that, about how it's correct, and you can expect that later. Until then, I hope you enjoyed this little video segment, and let me know what you think. I hope you found it helpful. Good night.